Hello students, welcome to EPG Pathshala. I am Dr. Pratibhanan, Principal from SP College of Pharmacy, Sonipat. Today we are going to discuss about the module Intraocular Drug Delivery under paper Novel Drug Delivery System Part 1. Now, there are many objectives. So I'll be discussing about the introduction, need for intraocular delivery, intraocular devices, under intraocular devices, liposomes, microparticles and nanoparticles, and ocular inserts. Now coming to the introduction, the caveats associated with systemic, topical or periocular delivery of drugs, that is, the poor intravitreal penetration results in ineffective treatment of posterior segment disorders. Topical administration of the drug is further limited by number of clearance mechanisms and the use of polymers with viscosity enhancing mucoadhesive properties to enhance the residence time has not yet been proved to be much efficient in the retention of drug in the vitreous. The chronic nature of posterior segment diseases thus require employment of alternative routes of administration which can provide sustained delivery of drug for a prolonged period of time. There is a need for intraocular delivery because the poor intravitreal penetration of systemically, topically or periocularly administered drugs, and secondly for the treatment of posterior segment disorders, and it requires the treatment for longer duration with sustained delivery of drugs. Topical delivery is limited by clearance mechanisms, and lastly it is the poor precorneal residence time. Intravitreal drug delivery. Intravitreal injections is an interesting route of delivery which minimizes the side effects related with systemic administration. An intravitreal injection requires a smaller volume of dose and delivers the drug adjacent to the desired site of action. Once injected, the drug diffuses through the vitreous gel and gets distributed throughout the vitreous humor, followed by a fast elimination. Drug is eliminated from the vitreous via two routes. Number 1, the anterior route where the drug diffuses to the posterior chamber, followed by removal to the systemic circulation along with the aqueous humor drainage. And second one, the posterior route where the drug is removed by active secretion across the retina. Drugs eliminated by the anterior route have a long half-life of 20 to 30 hours in a vitreous, whereas the drug lost by a posterior route, for example penicillins, they have a shorter half-life of 5 to 10 hours. Ocular inflammation, which causes the breakdown of blood retinal barrier, decreases the elimination of drugs that is removed by active transport and thus resides for a longer period in the vitreous. Since the posterior segment related diseases require treatment for longer duration with sustained delivery of active pharmaceutical ingredients, so colloidal particles like liposomes, microparticles or nanoparticles are found to be more suitable. Disadvantages of liposomes. There are a number of disadvantages of liposomes like the short shelf life and difficulty in storage. Another one is limited drug loading capacity and instability on sterilization. And lastly, it is the transient blurring of vision after an intravitreal injection. Dendrimers. Dendrimers are nanoscale, polymeric, highly branched, star-shaped, monodisperse macromolecules composed of three components. One is inner core, an inner shell of dendritic branches, and an external functional group. Dendrimers are basically synthesized from branched monomer units using two methodologies. First one is convergent and the second one is divergent method. The ability to attach the multiple functional groups makes them suitable for ocular drug delivery. Also, being artificially synthesized, it can be tailored with respect to size, shape, polarity, density, and solubility. Dendrimers having primary amine functional groups are referred to as full generation, whereas those with ester or carboxylate functional groups are called half-generation dendrimers. With increase in the number of branches, the dendrimers move from lower to higher generation. Dendrimers composed of polyamidoamine, that is PANAM, and have been commonly used in drug delivery as it offers high surface density functional groups like carboxylate, amine, and hydroxyl, making them suitable for conjugation of targeting ligands along with improving the solubility of a number of drugs. Dendrimers have been widely investigated for the delivery of ocular drugs such as pilocarpine and tropicamide. Pharmacokinetic study has shown improved bioavailability for both the drugs with significantly higher area under the curve as compared to phosphate buffer. The nature of the functional group present on the dendrimer may also impart bioadhesive property to it, enabling prolonged retention in the eye and improved bioavailability. Dendrimers have also been employed to transport drug molecules across the ocular tissues. To analyze the behavior of dendrimer on corneal permeation, a study was conducted by Yao et al. Using p-uranin with the aqueous solubility of 4 mg per ml as the model drug, where the dendrimer drug physical mixture and complex were compared. The results demonstrated sustained release of the drug with reduced burst effect by 25% with dendrimer drug complex. Permeability of dendrimer, however, was observed to depend on the molecular weight, size, and formulation technique. It was also observed that the physical mixture resulted in twice the permeability as observed by the complex. The reason for higher permeability was attributed to the ability of dendrimer to incorporate into lipidic bilayer of corneal epithelium, loosening the tight epithelial junctions, followed by release of drug from the dendrimer core and permeation through the epithelial intracellular space via paracellular route. Cationic dendrimers are composed of polyamidoamine, polyethylene, and have been used to incorporate and deliver genes, plasmid DNA or hairpin RNA to retinal pigmented epithelium and ganglionic cells of retina. Cationic dendrimers have been fabricated to deliver anti-VEGF oligonucleotides in rats to treat the choroidal neovascularization and was found to retain in the retinal tissue for 2 months, causing significant suppression in VEGF expression. Now coming to the liposomes. Liposomes are biodegradable, non-toxic in vesicular system composed of lipid membrane enclosing an aqueous core. So these vesicles are formed when the matrix of phospholipids is agitated in the aqueous medium to disperse in two phases. The commonly used phospholipids for the fabrication of liposomes are phosphatidylcholine, phosphatidic acid, sphingomyelin, and phosphatidylserine. Drugs encapsulated in the liposomal system are released in a sustained fashion, thus resulting in prolonged residence in the eye. They also reduce the intraocular toxicity of certain potent drugs such as antimetabolites, antivirals, and antibiotics to the retina. So, in a study comparing amphotericin B solution and a liposomal formulation, lesser toxicity was observed in the case of liposomal formulation. Liposomal have also been used to determine the integrity of the retinal vascular constitution by studying the distribution and release of dye loaded in the retina. Intravitreal injection of numerous liposomal loaded drugs in the rabbit models showed enhanced vitreal levels for extended period of time. Biodistribution studies of dexamethasone liposome showed the ability to bind to various ocular tissues such as retina, iris, sclera, and the cornea. Furthermore, the gold colloid loaded liposomes showed retinal attachment of liposomes to the inner lining of the membrane and its inability to penetrate the inner cells of the retina. Liposomes can therefore be used for site-specific delivery. Also, the heat-sensitive liposomes encapsulating the carboxyfluorosin have been used to study the potential of liposomes for targeting the drug to the specific area of retina. Liposomes, however, suffers from the limitations of shelf life, stability, storage, limiting drug loading capacity, instability on sterilization, and temporary blurring of vision after injection. So, despite these disadvantages, non-toxicity and biodegradability of these lipidic carriers make them highly suitable for ocular drug delivery. Microparticles and nanoparticles. These are colloidal particles of varying size range where the drug is either dispersed, encapsulated, or absorbed in the polymeric system. These particles are formulated by a number of methods such as emulsion polymerization, solvent evaporation, solvent diffusion, emulsification, solvent evaporation, precipitation. Biodegradable polymers are generally preferred for the ocular drug delivery due to the ability of these particles to retain within the eye, and it results in sustained release of drugs for longer time period. Microspheres are used for sustained delivery of retinoic acid in proliferative vitreoretinopathy. Microspheres are composed of biodegradable polymers containing ganciclovir. When it is placed intravitreally into the rabbits and inoculated with HCMV, it showed significant decrease in vitritis, retinitis, and optic neuritis. Targeting the potential of microspheres to the retinal pigment epithelium studied by using gelatin-coated microspheres showed enhanced uptake by the retinal pigment epithelium. Nanoparticles. Nanoparticles for the ocular drug delivery can be fabricated using number of polymers. Albumin nanoparticles can incorporate both positively as well as negatively charged drug molecules. Indomethacin-loaded chitosan-coated polycaprolactone demonstrated two-fold increase in ocular bioavailability, whereas the polyethylene glycol coating resulted in increased corneal permeability. In another study, chitosan sodium alginate encapsulating getafloxacin demonstrated sustained release for 24 hours after an initial burst release during the first hour, thus reducing the frequency of administration. Intravitreal delivery through suitable for the treatment of posterior segment diseases is a potentially painful route of administration, and repeated intravitreal administration are associated with risk conditions such as clouding of the vitreous humor, retinal detachment, and endophthalmitis. Coming to the intraocular devices. Intraocular devices are specially designed devices to provide sustained delivery of drugs to the eye for treating chronic intraocular infections by avoiding the undesirable side effects of the systemic route and multiple intravitreal injections, which carries a risk of infection. The introduction of implants, therefore, has significant potential in overcoming the above-mentioned limitations and therefore is a rapidly growing area in the field of ocular therapeutics. Now, implants or inserts are sterile, thin, multi-layered, drug-loaded, solid or semi-solid devices which are placed into the cul-de-sac and provide localized, controlled drug release over an extended period. So these devices are placed under a minor surgical procedure, making a small incision at pars plana, which is located posterior to the lens and anterior to the retina. Implantable ocular devices serve two major purposes. First one is the zero-order drug release rate, thereby improving the selectivity of drug action. And secondly, the release of drug over several months, thus reducing the frequency of administration. The desired criteria for any insert is ease of handling with ease of insertion, lack of expulsion during the wear, reproducibility of release kinetics, that is zero-order drug delivery, applicability to variety of drugs, non-interference with the vision and oxygen permeability, sterility, and ease of manufacture. The implantable devices are generally are of two types. Number one is erodible and another one is non-erodible. Now, coming to the classification of ocular inserts. Ocular inserts are classified into three categories. Number one, insoluble inserts. Secondly, the soluble inserts. And the third one is bioerodible inserts. Insoluble inserts include diffusion-based like Ocular, osmotic-based, and soft pre-soaked contact lenses. The soluble inserts include SODI, Biocorp, whereas the bioerodible inserts include the Lacrisert and Minidisc. Ocular was developed by ELSA Corporation. It is flat, flexible, wafer-like insoluble implant designed to release pilocarpine at a rate of 20 or 40 microgram per hour for a week to treat chronic glaucoma. It consists of three layers, that is two outer layers of ethylene vinyl acetate and an inner core of pilocarpine gelled with alginate. An annular ring of ethylene vinyl acetate impregnated with titanium dioxide for visibility enclose the inner core of pilocarpine. Lacrisert. It is another marketed intraocular sustained release device approved for treating cytomegalovirus retinitis. It consists of a pellet of ganciclovir, that is 6 mg, and coated with ethylene vinyl acetate and polyvinyl alcohol, and is designed to release ganciclovir at a rate of 2 microgram per hour for up to 32 weeks. This rate can be further tailored by varying the amount of polyvinyl alcohol and ethylene vinyl acetate. Now the next one is the Retisert. Retisert is an FDA-approved intraocular device which is used for the treatment of chronic uveitis affecting the posterior segment of eye. It is a silicon-laminated polyvinyl alcohol implant which is designed to release fluocinoacetonide in a sustained manner for up to 3 years. It helps to reduce uveitis recurrences, control the inflammation, and improve vision acuity. The associated side effects are cataract and elevated intraocular pressure. Now coming to the disadvantages. Number 1, have to be surgically implanted and removed after drug depletion. Secondly, it is too expensive. Another one, the non-compliance by the patients. Next, loss of device without being aware of the loss. Then sensation, that means it gives a sensation like a foreign body. Expulsion from the eye. People feel they find difficulty in handling an insertion. There are number of adverse events which are also reported along with this, like endophthalmitis, pseudo-endophthalmitis, vitreous haze, hemorrhage, cataract development, and retinal detachment. Pre-soaked contact lenses. These lenses, they are the hydrophilic lenses which are thin, curve-shaped plastic disc which adheres to the tear film over the cornea due to the surface tension. Numerous drugs like beta blockers, antihistamines, and antimicrobials can be delivered through these lenses. Contact lens provides longer residence and higher drug flux through the cornea with less drug inflow into the nasolacrimal duct. Drug is incorporated into these lenses by soaking them in drug solutions. It is found that most of the drug is released within the first 30 minutes. This method, however, results in inadequate drug loading and short-term drug release. Also, the use of preservatives may result in toxic effects on corneal epithelium. The other type of contact lens are particle-laden contact lenses where the drug is first entrapped in the carrier, such as liposomes, nanoparticles, or microemulsion, which is then dispersed in the contact lens material. Drug present in these lenses are released for up to as long as 180 hours and is therefore highly promising for extended ocular drug delivery. Unlike pre-soaked lens, it does not contain preservatives and are therefore non-toxic to the cornea. The lens should be kept in drug-saturated solutions to avoid drug loss during storage. However, this can be overcome by using the stimuli responsive, that is pH or temperature responsive particles, which will release the drug on response of the stimuli. Overcome the limitations of non-erodible implants. Erodible implants composed of biodegradable polymers were investigated. These implants are biocompatible and have sustained drug release properties similar to the non-erodible ones. However, they do not require to be surgically removed. The commonly used polymers in the fabrication of these implants are polylactic acid, that is PLA, polyglycolic acid, that is PGA, PLGA, and polycaprolactones. Till date, a few marketed erodible inserts have been developed, such as Soluble Ocular Drug Insert, that is SODI, Lacrisert, Minidisc, Surodex, and Ozudex. Soluble Ocular Drug Insert, that is SODI. It is a small, sterile, water-soluble oval wafer composed of soluble synthetic polymers, acrylamide, N-vinyl pyrrolidone, and ethyl acrylate weighing approximately 15 to 16 milligram. When wetted by the tear film, it softens in 10 to 15 seconds. Following 10 to 15 minutes, it turns into vitreous polymer mass, which after 30 to 60 minutes become a polymeric solution. A single application of this implant replaces 4 to 12 eye drops instillation or 3 to 6 applications of ointments. It is therefore presents a once-a-day application for the treatment of glaucoma. Lacrisert. It is a sterile rod-shaped device composed of hydroxypropyl cellulose and is used for the treatment of keratitis sicca and a dry eye syndrome. It weighs 5 mg with a diameter of 12.5 mm and a length of 3.5 mm. It is inserted into the inferior fornix and imbibes water in the conjunctiva and cornea, forming a thin hydrophilic film. It provides a day-long relief when placed in the eye in the morning. Minidisc. It is made up of contoured disc with a convex front and a concave back surface in contact with the eyeball. It measures 4 to 5 mm in diameter and is composed of silicon-based polymer. Minidisc can be hydrophilic or lipophilic, thus helping in incorporation of both water-soluble and water-insoluble drugs. Sulfasoxazole, a lipophilic drug, when incorporated in a hydrophilic matrix, showed drug released for up to 170 hours. However, gentamicin, when incorporated in a hydrophobic matrix, showed a release for 320 hours. Thurodex. Thurodex is composed of PLGA and hydroxypropyl methyl cellulose incorporating dexamethasone. It is placed in the anterior chamber of the eye to control post-operative inflammation in cataract patients. It provides sustained release of dexamethasone for a period of 7 to 10 days with improved anti-inflammatory effect comparable to topical steroid administration. Another one is Ozurdex. Ozurdex is an FDA-approved, biocompatible and biodegradable intravitreal device used for the treatment of macular edema. It utilizes Allergan's NovaDue technology and consists of PLGA polymer matrix and degrades slowly to lactic acid and glycolic acid and allowing the prolonged release of dexamethasone for up to 6 months. The Corneal Collagen Shield. This shield is a disposable, short-term therapeutic bandage lens for the cornea and composed of bovine collagen with a diameter of 14.5 mm, 9 mm of base curve and a thickness of 0.15 to 0.19 mm. Collagen is a structural protein that is used for various surgical and cosmetic purposes. It conforms to the shape of the eye, protects the corneal surface, and provides lubrication as it dissolves. Collagen shields are mainly used for the purpose of wound healing and dry eye syndrome. In a study, it has been observed in rabbits. The collagen shield impregnated with gentamicin showed the maximum concentration of the drug in the tear film and the tissue when compared to the eye drops, ointments, and conjunctival injection. These shields are sterilized by gamma radiation. However, the disadvantage is that the collagen shields are not optically clear and may cause some discomfort. Collagen shields are generally prepared by extracting the collagen and then molding into contact lens configuration. The drug is incorporated in the collagen shield at the time of manufacturing, and as the collagen dissolves, the drug is released in the tear film and corneal surface, increasing the permeation through the cornea and into the aqueous humor. Till date, number of drugs such as tobramycin, prednisolone, cyclosporine have been presented in the form of collagen shields. So now students, let us summarize what we have learned in this particular module. Intraocular drug delivery devices, they are used to provide therapeutic concentration of the drug at the intended site with a smaller dose and with improved bioavailability.